Now, the first speaker is Dr. Nicole Chen. Dr. Nicole Chen has experiences in the chemistry and nutrition of polysaccharides and proteins. She has a PhD in potato chemistry and nutrition. Her PhD investigated the relationship between the structure and functionality of whole potato tubers during processing. She has been working as a bioprocessing scientist at NanoLayer Limited since 2019. She's working on optimizing the electron spinability of microbial polysaccharides and marine collagen. Her passion is to bridge science, innovation, and industrial applications. She's going to talk to us about the healthier potatoes. Thank you, Dr. Nicole Chen, for musical presentation. The next speaker is Dr. Judy Duarte. Dr. Judy has a Bachelor of Food Engineering and a PhD in Pharmaceutical and Food Sciences from University of Antioquia in Medellin, Colombia. She has experiences in leading research, development, and innovation projects related to the dehydration, quality, preservation, and fortification of foods. The outcomes of these projects have been published in international journals and communicated at national and international scientific congresses. She is currently a professor in the Department of Food and a researcher of the BioAli Research Group at the University of Antioquia. She also acts as a project manager and coordinator of the Intel Research Group at the Institute of Food Science and Technology, Intel in Medellin, Colombia. The title of her presentation is Development of Fortified Low-Fat Potato Chips Through Vacuum Impregnation and Microwave Vacuum Drying. Off to you, Dr.
Do you like potato chips? Do you know that potato chips are the most widely consumed snack in the world? The problem is they are traditionally prepared by conventional frying, and they are known to be a significant source of fat intake. Their fat content ranges between 33 and 38%. However, today consumption is changing towards healthier alternatives. What if I tell you there is a way to eat potato snacks with less fat content? In addition, potato snacks can be used as an ideal strategy for nutrient delivery due to their high consumption worldwide. My research aims to produce potato chips using the hydration technology microwave vacuum drying, previously fortified using the vacuum impregnation technique. In the first part, potato slices were submerged in a fortifying emulsion containing vitamin C, vitamin E, and calcium, and subjected to a vacuum impregnation process. It has been described as a fast mass transfer phenomenon and consists of exchanging the internal gas or liquid occluded in open pores for an external liquid phase promoted by the pressure gradients in the system. We have already the fortified potato slices. Now it's time to prepare the dehydrated potato snacks using microwave vacuum drying. It is a dehydration process that uses microwave radiation to generate heat in a chamber with absolute pressure lower than atmospheric pressure. It allows a short drying time, about 20 minutes, and produces high-quality materials. After the drying process, a composition analysis could determine that 100 grams of fortified and dehydrated snacks potato chips had 37 milligrams of vitamin E, 280 milligrams of vitamin C, and 890 milligrams of calcium. Regarding the fat content, it had only 0.2% of fat. Now we can all enjoy healthier snacks.
Thank you, Dr. Yudi, for a very interesting talk. The next speaker is Dr. Setya Abdul. Dr. Setya Abdul is an assistant professor at the Laboratory of Food and Agricultural Products Engineering in the Department of Agriculture, Faculty of Animal and Agricultural Sciences, Universitas Diponegoro in Semarang, Indonesia. He has the Bachelor of Science and a Master of Science from Gadjah Mada University in Indonesia, the Master of Science from University of Hohenheim, Germany, and a PhD from University of Otago, New Zealand. He has extensive experiences in working with meat, dairy, and most recently, potato products. His research interest is in the area of sustainable food processing. The title of his presentation is Kinetics of Color Development During Frying a Potato, Pre-treated with Post-Electric Fields and Benching, Effect of Cultivar.
Hello everyone, I'm happy today to share with you the work of our team from Universitas Diponegoro and University of Otago entitled Kinetics of Color Development During Frying of Potato, Pre-treated with Pulse Electric Fields and Blanching, an effect of cultivar. Pulse electric fields, or PEF, processing applies high voltage electric fields in the form of short pulses across biological cells or foods. PEF leads to pore formation in the cell membrane. PEF has been commercially used in deep-fried potato industry. One of its benefits is that the color of potato fries become uniformly bright brown. Color is a critical quality attribute of potato chips. The typical color of potato chips are yellow to brown, and its formation occurs during frying. Precursors such as reducing sugars and amino acids of potato are known to be responsible in the color development during frying. Potato chips that are dark in color is undesirable. Therefore, breeding of potato cultivar that is suitable for producing potato chips with desired color is still going on. This research was aimed to study the effect of PEF treatment on the color development of potato chips during frying, with special emphasis on the effect of potato cultivar. Four potato cultivars, namely Crop 77, Moonlight, Russet Burbank, and Nadine, were examined for their color development during frying with or without PEF treatment. Here it is the visually observed color of potato chips from the four potato cultivars without PEF treatment. As can be seen, the color of potato chips got darkened more quickly at a higher frying temperature. It is also shown that potato low in glucose, such as Crop 77, got darkened more slowly than that high in glucose, such as Nadine. Regarding the kinetics parameters, the order of reaction of color darkening of potato chips followed first-order reaction model. Furthermore, potato that is low in glucose had a low K value of color darkening, and vice versa for that high in glucose. As estimated using Arrhenius model for the temperature-dependent, PEF lowered the activation energy of color darkening. Thus, the change in K value became less temperature sensitive, or became more temperature stable due to PEF. As conclusion, we saw that regarding the color development during frying of potato slice, PEF improved the frying performance of potato cultivars that are suitable to produce potato chips. Here, I would like to acknowledge LPDP, FIAT, RIDET, and Plant and Food Research of New Zealand for different types of support upon this study. Thank you.
Thank you, Dr. Setya, for his informative talk about the color development during frying a potato. The next speaker is Dr. Mao Jen. Dr. Mao Jen is an associate professor in the Potato Processing and Quality Control Innovation Team within the Institute of Food Science and Technology of Chinese Academy of Agricultural Sciences in Beijing, China. Dr. Mao's research mainly focuses on the structure and functional properties of potato protein and its bioactive peptides. She obtained her PhD from Chinese Academy of Agricultural Sciences in 2012. She was a visiting scholar at Auburn University in the United States in 2018. She has published multiple articles in international journals of food hydrocolloids, food chemistry, and ultrasonic solar chemistry in the last 5 years. She's going to talk to us about contribution of high hydrostatic pressure to anti-proliferative effect of potato protein on HT29 colon cancer cells.
Hello everyone, my topic is contribution of high hydrostatic pressure to anti-proliferative effect of potato protein on HT29 colon cancer cells. Soluble proteins present in potato fruit juice have been divided into 3 groups. One of them is 30 to 40% patateins. Patatin presents various biological activities, such as anti-cancer, antioxidant activity, etc. Colorectal cancer is the 3rd most common cancer and the 4th leading cause of death from cancer worldwide. High hydrostatic pressure, a now-known thermal technology, can modify the structure of biopolymers, such as protein, thus may change the functional and biological properties of food protein. However, to our best knowledge, there was no report about the effect of HHP treatment on anti-proliferative activity of patatin on HT29 cells. So, HHP treatment and 250 to 550 MPa for 15 minutes was used to modify patatin. And we found that HHP treatment induced the increase in beta strands, random core, beta potential, the decrease in alpha helix of patatin, and improve its anti-proliferative effect. HHP-treated patatin and 450 and 550 MPa induced potent apoptosis and cell cycle arrest in G1 phase of HT29 cells. HHP-treated patatin also induced autophagy and DNA fragmentation in HT29 cells. In conclusion, HHP-treated patatin possesses inhibitory effect against HT29 colon cancer cells in vitro, which was attributed to the apoptosis and autophagy. That's all. Thank you.
Thank you, Dr. Mao Jen, for her inspiring talk. The next speaker is Dr. Mingmei Ma. Dr. Mingmei Ma obtained her PhD from Chinese Academy of Agricultural Sciences in 2016. She is currently an assistant professor in the Institute of Food Science and Technology of Chinese Academy of Agricultural Science, China. As a key researcher in the potato processing innovation team, she has been involved in potato processing and utilization of potato byproducts. Dr. Ma has published 26 papers, 4 agricultural industry standards, and 8 books. She also holds 7 national invention patents. The title of her presentation is Ultrasonic Microwave Assisted Acid Extraction of Pectin from Potato Residues and its Characterization.
Hello everyone, it's my honor to share the topic ultrasonic microwave assisted acid extraction of pectin from potato residues and its characterization with you. Maybe you will ask, why potato pectin was studied? You know, in China, potato is mainly used to process starch, and about 7 tons of fresh residues are generated from 1 ton of starch production. However, due to the lack of high-value utilization technology, about 80% potato residues were discarded, resulting in serious environmental pollution. It was reported that potato residues contained more than 17% pectin, so exploring the extraction method and clarifying the characteristics of potato pectin is urgent to improve the added value of potato residues. Therefore, we optimized the extraction parameters of potato pectin and investigated its structure and emulsifying properties. The optimum extraction conditions were ultrasound power was 50 watt, temperature was 93 degree, pH was 2, time was 15 minutes. The yield of potato pectin was more than 22% under pilot scale. The potato pectin exhibited linear apparent structure with short chains. Further, the RG1 and degree of acetylation was much higher than that of apple and citrus pectin, which was related to the better emulsifying properties. Next, we found that higher emulsifying activity and stability was observed when potato pectin concentration was more than 2.5%, oil phase was 50%, and pH was 5. In the future, we will introduce the extraction technology to more enterprises and achieve the industrialized production of potato pectin. Moreover, structure, stability changes, and physiological activities of potato pectin will be investigated in order to expand its application in food and health products processing. That's all. Thank you.
Thank you, Dr. Meng Mei Ma, for her interesting talk. The next speaker is Li Chu. Li Chu obtained her Bachelor of Food Science and Engineering and her Master of Food Science from Shanxi Normal University in China. She is currently a PhD student in the Institute of Food Science and Technology of Chinese Academy of Agricultural Sciences. Her research focuses on the high-value utilization of sweet potato residues. She has recently published her research paper in Journal of Food Chemistry. She's going to talk to us about nutritional composition, antioxidant activity, volatile compounds, and stability properties of sweet potato residues fermented with selected lactic acid bacteria and bifidobacteria.
Hello everyone, my report topic is nutritional composition, antioxidant activity, volatile compounds, and stability properties of sweet potato residues fermented with selected lactic acid bacteria and bifidobacteria. The sweet potato of China was accounting for 56.62% of world total production. Most of them were used to produce starch and their own products. In this process, a large amount of sweet potato residues generated and often discarded directly, resulting in serious resource waste and environmental pollution. Microbial fermentation of plant-based material contributed to its higher nutritional value, better taste, stronger antioxidant activity, and so on. How about the effect of fermentation on sweet potato residues? It is still unknown. The SPR were hydrolyzed with alpha amylase, and then 5 kinds of strain solution were allotted respectively to obtain fermented SPR. The nutritional composition, antioxidant activity, volatile compounds, and the stability properties of fermented SPR were evaluated. Microbial fermentation caused an increase in the content of soluble dietary fiber, total polyphenols, and organic acids. The flap capacity and DPPH radical scavenger activity was significantly improved after fermentation, of which the SPR fermented with LR showed the highest value. The fermented SPR showed a higher umami and sourness tastes, and the acid relative peak areas were increased significantly. The apparent viscosity of fermented SPR were higher than SPR. R squared is more than 0.95, indicating that this model could well describe the flow behavior of samples. The increased apparent viscosity contributed to the stability of fermented SPR system. This study provides evidence to support the fermentation was lactic acid bacteria and bifidobacteria, which can be a novel and environmental-friendly processing technology for SPR, and making it a great potential for the production of functional foods. Thank you for your attention.
Thank you, Lily Chu, for her interesting talk about the properties of sweet potato residues. The next speaker is Lulu Li. Lulu Li is currently a PhD student in the Institute of Food Science and Technology of Chinese Academy of Agricultural Sciences. She obtained her Master of Food Science also from Chinese Academy of Agricultural Sciences. Her research focuses on the storage of agricultural products. She has recently published her research papers in journals of ultrasonic sonochemistry and LWT Food Science and Technology. The title of her presentation is Effective Ultrasound Combined with Slightly Acid Electrolysis Water on an Activating Rhizopus stolonifer in Sweet Potato.
Hello everyone, my name is Li Lulu. Today I would like to share my academic research findings under the title Effect of Ultrasound Combined with Slightly Acid Electrolysis Water on an Activating stolonifer in Sweet Potato. My research work is divided into 4 parts. Sweet potato has a wide planting area in high yield in China. According to Food Agriculture Organization, China ranks first globally in sweet potato production in 2019. However, sweet potato tuber is easily infected by various fungi, which affect sweet potato storage time. The most serious disease is the soft rot of sweet potatoes, as you can see from this figure. At present, post-harvest control of sweet potato mainly includes 2 aspects. The first one is the screening of disease-resistant species. However, different studies show that no species have shown completely resistance. The second aspect is the use of traditional fungicides, but it will lead to harmful substance remaining after use. Therefore, there is not a need to find growing preservation methods. Our methods are summarized in this chart, whereby we aimed at finding novel physical preservation methods to inhibit the growth of fungi on sweet potato saplings in order to prolong the storage time. Ultrasound and hot water treatment inhibit methylene growth and spore germination. Besides, ultrasound and hot water treatment inhibit high-end fungal activities against R-stolonifer. Ultrasound combined with slightly acid electrolysis water treatment could control the growth of R-stolonifer in sweet potato tuber roots to some extent, indicating that it could be a new way to control R-stolonifer in sweet potato. Ultrasound combined with slightly acid electrolysis water treatment can maintain the physiological quality and nutritional value of sweet potato during storage. We concluded that the shelf life of sweet potato can be prolonged successfully by ultrasound combined with slightly acid electrolysis water. Thank you for listening.
Thank you, Lulu Lee, for your informative presentation. Moving on to our next speaker, Dr. Yadunath Bagai. Dr. Yadunath Bagai has worked in different positions within the Ministry of Agriculture and Forest in Bhutan in the last 23 years. His current role is National Potato Coordinator in the National Center for Organic Agriculture in Yusipang, Thimphu, Bhutan. He has an academic background in agriculture. He has a Bachelor of Science from Bengal University in the UK, Master of Science from Chiang Mai University, and PhD from the University of New England in Australia. He has authored and co-authored more than 50 national and international publications. His presentation is titled Rising Role of Potato in Bhutanese Diet.
Greetings to everyone from Bhutan, and thank you for inviting me to this international symposium. I would like to talk on rising role of potato in Bhutanese diet. Potato is grown in mid and high altitude regions of Bhutan. Green and blue areas are where potato is mainly grown on. Potato contributes to food security in Bhutan because it is eaten as vegetable and even as staple food. Cash generated through sale of potato is used to purchase rice where rice is not grown. Potato has acted as catalyst to transform the subsistence agriculture to market-oriented economy. We all know potato contains mainly carbohydrate, protein and micronutrients. It is 80% water. Consumption in Bhutan. In Bhutan, potato is widely eaten as kewadachi, a local item with cottage cheese chili potato. It is used as baked, boiled French fries or processed chips. It is also eaten as kewa pakoda, as vegetable or in other forms. Consumption in Bhutan is rising. In 1970s, per capita consumption was just over 5 kg per person per year. In 1982, it rose to 20 kg, and in 2006 to 2019, it is about 26 kg per person per year. That works out to be about 71 gram potato per day per person, which contributes about 47.47 kilocalorie energy per day and 1.13 gram protein and 0.71 gram fat. Because of its importance in Bhutanese economy and diet, we have introduced new germplasm from International Potato Center in Peru, and we have released a new variety called Usimap in 2017. It is high yielding. It has moderate resistance to lead blight. Good thing about this variety is that it is micronutrient dense. It has significant quantity of iron, zinc and vitamin C, which we think will solve some of the issues with malnutrition in our youth, young ladies, and general consumers. Because of this merit and late blight resistant, high yielding traits, it is very popular with our farmers, and that is how this variety has been featured as cover page in annual publication of Ministry of Agriculture in Bhutan. So we are going to introduce more varieties for farming system. Thank you.
Thank you, Dr. Yaduna, on his talk, for his talk on the role of potato in Bhutanese diet. Our last speaker is Katia Bravo Bendezu. Katia is originally from Peru and is currently residing in the United States. She's working in quality control for Clio Snacks Company in New Jersey. She has a Bachelor of Food Science and Engineering from San Cristobal of Huamanga University. She's currently completing a master's degree at the Faculty of Food Science and Technology of National Agrarian University La Molina in Lima, Peru. She has more than 10 years of extensive experience in quality assurance, working in the food industry and several universities in Peru. The title of her presentation is Influence of the Amylose Content on the Functional Properties of Starch of 6 Native and Modified Potato Cultivars.
Good afternoon, my name is Katia Bravo. I'm going to talk about my research on starch potatoes. Peru is the first potato producer in Latin America, but the starch industry is small. Understanding the physicochemical and functional properties of these starches could be useful for food industry application. The objective of the research was to determine the possible influence of amylose on the functional property of starch isolate in 6 potatoes, native and improved varieties. Potatoes were provided by SIP. In this way, the environmental factor was controlled. On the other hand, all starch potatoes were classified as having low phosphorus content and didn't show significant difference among them. This is a description of the potato samples. 2 methods, colorimetric and enzymatic, were studied to determine the amylose content in potato starches. The enzymatic method was selected. Amylose content values of different starch ranged between 21 and 25%, and therefore all starch potatoes were classified as normal amylose content. The thermogram for starch potatoes showed the lowest thermal profile corresponded to the CYP001 variety. Researchers reported that the amorphous amylose in the normal potato starch decreased the relative amount of crystalline material in the granule, thereby lowering the gelatinization parameter. Pasting. Viscograms of starch pasta showed that CYP006 was the sample with the greatest peak viscosity and presented the highest breakdown viscosity. The peak viscosity is related with the granule starch swelling, which is mainly a property of amylopectin. Rheological property. All these starches formed gels with a pseudoplastic gut behavior. CYP003 was the strongest gel, while CYP001 was the weakest gel, possibly related to its thermal story and the loss of its granular integrity. The Pearson correlation showed that amylose had a significant positive correlation with the setback. Also showed that gelatinization temperature had a significant negative correlation with the phase angle, but a significant positive correlation with the hot pass viscosity and the final viscosity. In the same way, the phase angle had a significant negative correlation with the mass temperature, the hot pass viscosity, and the final viscosity. For the amylose, the recovery of viscosity during cooling was mainly attributed to amylose content. In the second case, the starch granules, mainly composed of amylopectin, have also influenced the pasting and the rheological properties, affecting even the firmness of the gel. Some studies indicate that gelatinization temperature is related to the quality of amylopectin, and the quality of amylopectin is related with the fine structure of amylopectin. In conclusion, for the starch studied, amylose in the studied ranges influenced the setback viscosity. The gelatinization temperature related to the amylopectin influence on the swelling of the starch granules and therefore influenced the thermal, viscoelastic, and rheological properties of the starches.
Thank you, Katia. And I would like to congratulate all of our young scientist speakers for their wonderful and informative presentations.